bonding interactions between the amino acids. So one of the most important ones is hydrogen bonds. So you can have like intramolecular hydrogen bonds between like large molecules, but for amino acids, we know it as a strong intermolecular force between the different residues. So hydrogen bonds, they're created because of the differences in electronegativities between hydrogen and a electronegative group such as oxygen, nitrogen, or fluorine. And in amino acids, we are particularly interested in the nitrogen and the oxygen, since there are no fluorines on the amino acids. So because of the electronegativities, the hydrogen has a partial positive charge on it, while the oxygen or nitrogen has a partial negative charge. That's because the electronegativity of the oxygen nitrogen, it draws more of the electron density towards the oxygen or nitrogen, which leaves a partial positive charge on the hydrogen. So because of the partial positive charge on the hydrogen, it is able to interact with the partial negative charge of a oxygen or nitrogen on another amino acid. So as you can see in the picture on the right, because of the partial positive charge on the hydrogen will interact with the oxygen or nitrogen on another molecule that has the partial negative charge. And so in that case, we have a donor that donates the hydrogen for the hydrogen bond. So it's any molecule with a oxygen, nitrogen, or fluorine that has a hydrogen bonded to it that creates the partial positive charge on the hydrogen. And then acceptors are the molecules with either an oxygen, nitrogen, or fluorine with a lone pair of electrons that has the partial negative charge, and that binds to the hydrogen from the donor. Yeah, so in amino acids and proteins, the amino group will hydrogen bond with the carboxyl groups in the peptide backbone, and that forms the secondary structure of proteins. So the primary structure is the long chain of peptides that forms the polypeptide chain, but the hydrogen bonds between the amino and carboxyl groups of each amino acid, the interactions between them, between the different residues, they form the secondary structure of proteins. So the two different types on the MCAT are the alpha helices. On the left, you can see it's that spiral shape, and then the beta pleated sheets, which are the sheets that lay on top of each other, well, side by side in this picture. And the hydrogen bonds, they provide stability to the protein. So the hydrogen bonds, they form the proper secondary shape of the protein, and it provides stability until like a denaturant comes by and breaks the hydrogen bonds. The other interactions by amino acids are the hydrophobic interactions. So like I said before, the different hydrophobic amino acids, they'll be attracted to each other because they're trying to repel the aqueous environment. So they'll try to repel the polar molecules such as water or the other hydrophilic amino acids. So as a result, the hydrophobic amino acids will be on the inside of a protein. So they'll be in the interior of their core, and this allows more hydrophilic amino acids to interact with the aqueous environment, because the hydrophilic amino acids, they'll be pushed to the outside, the exteriors of the protein, while the hydrophobic amino acids are able to become concentrated in the interior core of the protein. So the hydrophobic amino acids will interact with each other while pushing out the hydrophilic amino acids to the outside for it to interact with the aqueous environment. And this maximizes stability of the protein because it won't be like a huge mess of random hydrophobic and hydrophilic amino acids interacting with each other. It becomes more concentrated where the hydrophobic amino acids are on the insides of the protein while the outsides are more hydrophilic interactions. And hydrophobic interactions, this determines the tertiary structure of proteins. So this forms the tertiary structure of proteins, which is usually the final form of the protein, unless it has a quaternary structure. And so on the other hand, there are hydrophilic interactions where the hydrophilic amino acids, they're found, like I said, on the exterior of the protein. And this is this is done in order for it to interact with the polar and aqueous molecules in the environment. So most proteins, they work in the cytoplasm or other aqueous environments. And as a result, the hydrophilic amino acids are on the outside because it shields the hydrophobic residues from the aqueous environment. So like I said, this will concentrate the hydrophobic amino acids in the center, while the hydrophilic ones are on the outside, and this is done to increase stability of the protein. So since the hydrophilic amino acids are on the exterior of the protein, it creates what we call a solvation layer around the protein. So this is a layer of water molecules, and this is done to increase hydrogen bonding. So the water is forced into a specific arrangement that maximizes hydrogen bonding with the hydrophilic amino acids. And this also contributes to the tertiary structure of proteins.